All right, now at this point, if you're wondering why you see the writing but you don't see me, that's because I am wearing all black so I blend in with the background. Now, we're getting near the end. This is the last section, so we're almost through, ladies and gentlemen. Okay, so typically, under normal circumstances, or in most cases, many plants are referred to as C3 plants. That's because the initial molecule that's made with the fixation of carbon carbon dioxide is a three carbon molecule. So typically we have CO2 using the enzyme Rubisco that to eventually form these organic molecules. Now, there are certain situations, though, in which access and concentrations of carbon dioxide will change. Now, if we think of the mesophyll of a leaf, and we look here, and there's the palisade mesophyll, and the loose mesophyll, spongy mesophyll. If it is a particularly hot and dry day, then there will be instances when the stomata that allow gases to flow, carbon dioxide to flow in, oxygen to flow out, will close completely or partially, and that will prevent gases like CO2 from being able to enter. Now, CO2 is the molecule needed to make those sugars. So what will happen is gas concentrations within the chloroplast will drop. Now, looking at Rubisco a little bit more closely, Rubisco typically bonds with CO2. Now, when you have a high concentration of CO2, Rubisco joins with it, and RUBPH, and you're off and running, you're making sugars. Now, if you have a situation in which the stomata are closed, carbon dioxide levels will begin to drop, so there's a less readily available source of carbon to make sugars. In addition to that, the light reactions are still happening, so water is still being split apart and being used to make oxygen. So what will happen is the oxygen levels will actually, oxygen concentrations will actually begin to increase. Now, when this happens, Rubisco will alternatively bond with oxygen and create an intermediate compound. Ooh, let me just clean that up there a little bit. So it creates an intermediate compound that will eventually then be used to produce the CO2 needed to make sugars. Now, this process is called photorespiration. Photo because it uses light or energy from light, and respiration because it produces CO2. Now, the kicker with photorespiration is it actually uses ATP and ultimately produces no sugars. Now, when this happens, when photorespiration begins to occur, sugar production will actually drop in plants. So you get less output from plants as they go through photorespiration. The thought behind this, though, the evolutionary purpose of this process is the photoprotective role of photorespiration. Now, the thought is that by going through this process, prevents the buildup of products from light reaction that could be damaging to the cell. Now, looking at this from the viewpoint of another category of plants, we can look at C4 plants. Now, C4 plants get their name because, again, the first compound made is a 4-carbon compound. Now, these particular plants are or have evolved to be adapted to hot, arid, or dry climates. Now, in such environments, plants need a way to disconnect the absorption of carbon dioxide and oxygen with the production of sugars. And C4 plants do this structurally. So if we can imagine looking at a cross-section of a leaf, we can envision the veins of that leaf. So they're actually, think of it as plumbing in the leaf that brings in water and pumps out sugars. So that's the plumbing of the cell. And surrounding that vein or that plumbing are cells called bundled sheaths. And then surrounding the bundle sheaths are mesophyll cells. Maybe give those slightly different colors so they stand out a little bit more easily. Okay, so we have the bundles. And the mesophyll. Now, in sort of envisioning the relationship between these processes, we can take, oops, I'll give you a little bit more space here to write. We can take these mesophyll cells and take carbon dioxide and have that carbon dioxide join with a molecule abbreviated PEP. Now, what's interesting is that PEP, this PEP molecule, can combine with carbon dioxide using an enzyme called PEP carboxylase. Whew, spelled it. Now, what's great is that PEP carboxylase can fix carbon dioxide even when CO2 concentrations are very low. So even when the stomata are closed, PEP carboxylase can take this CO2 that is available and combine it with PEP to create this 4-carbon intermediate, hence the C4 categorization. So you have a 4-carbon intermediate. Now, remember the mesophyll cell is adjoining a bundle sheath cell. And when the 4-carbon intermediate passes through the plasmodesmata into the adjoining bundle cell, our bundle sheath cell, what occurs is pyruvate, that 3-carbon molecule, is split off, and carbon dioxide is released and able to enter the Calvin cycle. So what C4 plants have basically done to adapt to this hot, dry environment is to have adjoining cells that contain an enzyme that allows CO2 to get fixed into 4-carbon intermediate even when CO2 levels are low. That intermediate then passes on to the bundle sheath cell where the carbon dioxide is again split out and used to make the G3P sugars that we're familiar with as products of the Calvin cycle.